Пускай Тимур достанет свои слайды, потому что, может, это займет какое-то время.
Да. Тимур, а ты хочешь чего-нибудь еще добавить к какую-нибудь общую информацию, которая...
Ам, Наверное, единственное, что вот данная моя позиция сейчас, я postdoctoral fellow at Keio University in Tokyo. И извините, я плохо слышу.
PhD ты в Вайцмане делал?
Да, PhD я получил в Weizmann Institute of Science, Израиль. Вот, а до этого, получается, Masters в Moscow State University в Москве. И тема, то, в чем я заинтересован, как было объявлено, действительно, микробиом, я so as we start, I want to switch into English. It's easier. I apologize. But so, in fact, I wanted to structure the lecture today not as a academic lecture, but more as a informal conversation. This way, we will avoid. We don't have fixed goals to reach, and therefore, and any questions that come up at this audience, we can address to the best of my knowledge and the extent of my knowledge, and this way we can balance the scientific accuracy on one hand, and also the accessibility of information to not specialists, which is of course the point, and what I would like to do. So I would like to start with a little bit far from the specific topic of today's talk. That is that it's what fascinates me about science is that you can see patterns that hold true for things that seemingly are not connected. For example, the way wind moves molecules of air, in weather, is is described by the same exact laws that also describe how the internal combustion engine works. And in a similar manner, rules of ecology that we now see, governing climate change and modern extinctions of species, and we see consequences of reduction in diversity and stability of ecosystems. And we now, we as a society, as a species, now becoming more aware that the ecosystem is not bulletproof, and that, for example, if tomorrow the bees will go extinct, in a few years, 90% of human population will also go, and there is nothing we can do about it. Or that if we, when we use monocultures, for example, like banana, what happens, inevitably a predator arises that can completely disseminate or destroy the monoculture. Monoculture essentially are extremely vulnerable, and the bananas that we eat now today are not the bananas that our grandparents ate, because that wasn't the dominant culture. Actually, the banana went extinct already 3 times, and now we're in the progress of the 4th extinction because of the fungus. So every time we decide we choose a monoculture, a fungus arises, kills it off. We seemingly, hiding it from consumers, of course, switch the culture, another fungus arises, and so on. So the lessons are that diversity is important, and that parts of ecosystem depend on each other. And interesting, as I said in the beginning, patterns hold true on different in different settings, but and this applies also to scales. So on the macro scale, we know that we depend on the climate, we depend on bees, we depend on other parts of the ecosystem. But interestingly, the same is true on an individual level for each and every one of us. Each and every one of us represent an ecosystem in ourselves. That is because we live together with a number of microorganisms that come from several kingdoms, very diverse. Yeah, please.
Sorry to interrupt already, but I'm curious about your banana example. I understand you just refer to it, just to cite something which is supposedly well known, but I had absolutely no idea about banana extinction. Neither I knew about banana monoculture. So, from where I sit in Boston, it seems like we always have bananas. We always have plenty of different bananas. There's organic banana, and there's, there are different, what do you call those? Platanas or something like that, which are different. But yeah. Anyways, I'm just curious if you can tell a little bit more about this banana extinction.
Absolutely. And also in general, the reason why we don't have fixed slides today is exactly this. So please stop me. Anything that is not clear or interesting, we can go in that direction. So regarding the bananas, all bananas of the regular size that we usually call bananas is just one plant, one species. And this species is not going to be with us for much longer because of the see above. It is being ravished by fungus that we cannot stop. We don't know how to stop. It's a big industry, but no one can stop it. And the bananas that were on the market in the 50s and 60s it was a different species of banana. It was a Cavendish banana, and you can still buy it. There are still spots on the planet where you can get them, but the problem is that these spots can never get to scale, because the moment they get to scale, they get eaten, destroyed. So they can only survive as very small plants, very small plantations. And if you do try to order Cavendish banana in the US, it will cost you several $100s for a pack because it's super rare, and they are sweeter. And this is true if when grandparents say, specifically to about bananas, when elders tell us that things were sweeter and better, this is just true for the case of the banana. The previous dominant species on the market was just sweeter, fatter, and was a better crop. And
when you say that we decided to stick with the monoculture, you don't really mean some kind of a worldwide banana federation. You just mean that it sort of was selected as a dominant species, agriculturally most favorable, right?
Yeah, of course. So there are many, it's natural selection essentially, that the crop that requires less water, less care, less of everything wins out, and that becomes dominant as the main culture. So that's what I'm talking about.
Thank you.
Sure. So regarding the so essentially the same lessons that apply to diversity, stability of ecosystems on a macro scale that we live inside of also apply to ourselves. And the revelation of the last 15 years is that there is this organ that pretty much no one looked at in our body that is crucial for our health, for our immune system, for our metabolic health, for our mental health as well, and that is the microbiome. And the microbiome is from the name is the collection of all microorganisms that are associated with us. And we understand that there are thousands of species of bacteria, many more viruses that are associated with us, also like simple single cell animals and archaea as well. How do we know that they are crucial to our health? This audience most likely doesn't need a reminder about the difference between observational studies and causation studies, but just to go briefly through this, of course, we're most interested in human health, but of course, for ethical reasons, logistical reasons, because our lifespan and other things, we can only do so much with studying ourselves, humans. And when it comes to causation, proving and showing causation that something actually directly leads to something, it's very, very difficult in humans, and we wouldn't be where we are in terms of lifespan, medical care, and everything if we wouldn't perform animal experiments on essentially modeling humans using animals that are more useful for studies in terms of logistics, lifespan, and so on. And this introduces a grain of salt to any results that we get from animal studies. And so we need to structure our questions that we ask in animal studies in a way that would be translatable to humans. So we need to, for example, we basically need to ask very fundamental questions that are for sure will be applicable to us as well. And also, we probably don't need a reminder of what a gene is. Gene is the element of hereditary or evolution, and the sorry yeah, we discussed yeah, so this is justifiable on this level as well, because as you might know, we and the most used animal model, rodents, mice, rats, are 97% identical to us in terms of genome, and two randomly picked humans on Earth are 99.9% identical in terms of genome. And so on this basis, we can say that we can use rats and mice for the animal studies. With regards, I also want to go through several other terms that we're going to use discussing microbiome. For example, sequencing. Sequencing is a process of determining the structure of, or the structure of the gene. And when we talk about the diversity or the composition of the community of microbes that live on us, one might ask, how do we know? How do we know what's there? And also, earlier I mentioned that the revelation about 15 years ago about the role of this organ. You might also ask why 15 years ago? And the reason is the convergence of technologies that allowed us to sequence bacterial DNA very, very cheaply. And essentially, this led to the method that is now being used to look at diversity of microbes. So the way we look at diversity of microbes, or we know what's there, is that we collect a sample and then sequence all the DNA in it, and then basically align or look up each sequence to see if it matches any known organism that we know of, and that's how we get the composition of microbes in any given sample. Why this was such a big game changer? That's because living organisms like us represent a dynamic and changing environment for microbes. So it's very difficult to recreate independently from the living organism. So how did microbiology, how did we study bacteria before? Before, to study bacteria, you had to be able to grow them in in a media or on an agar plate or somehow. And so for the most part of history of microbiology, all of the methods were culture dependent. But now, as I mentioned, we don't have to culture anything. We can just scoop up the sample and just look what's there. We not only can look at who is there, but also we can look at what they are doing collectively, what kind of biological processes are going on. We cannot yet pinpoint what biological process is connected to which bug, but it's getting there.
Timur, I'm sorry. May I ask you, are you using human cell culture, let's say intestinal cells or intestinal organoids to study microbiome? Because those are human cells from intestinal tissue or from colon that you can grow and 3D, for example, and look how it works. And you can also use immune cells there or some other cell types. Or is it still not a method that is currently used to study microbiome?
Absolutely, absolutely. We do use for certain applications. So there are immortalized lines. Essentially, there is a way to make any cell that we take out of our body immortal, basically cancerous. It will reproduce endlessly. And this is what's called cell line. And they are useful, for example, for drug screenings. And now we are at the point where many laboratories, many pharmaceutical firms are trying to model the gut environment for all the test and screening purposes. So, but mostly when it comes to causation, in vivo studies are the ones that are, so the studies where the live animal models are used. We do use cell lines, and here we mostly do this to look at the mechanism, because so let's say we observe some phenomena. Let's say we observe that some bacteria is doing something in a live mouse, but then we want to know how does this bacteria exactly does what it does, and then even the lifespan of a mouse becomes too long, and it's too difficult logistically to work with it, and the question doesn't require it. And if we, for example, have identified that bacteria is interacting somehow, for example, with an epithelial cell of the lining of the gut, then it makes sense to actually take this cell out, make a cell line, or buy it, and then study the interaction between them separately from the organism, because we have now selected and identified our question. So absolutely, we do use cell lines, and yeah.
Do you know that you can use primary derived normal cells now because they have contain some stem cells, and so you can still grow them? as a small models, like in 3D, like
you mean
mini organ, basically.
You mean organoids?
Organoid,
Organoid systems. Yeah. Absolutely, yes. That's another. Wow. So yes. So there is another step between the cells, immortalized cell lines and live animal models, and this is indeed organoids. They have limitations as well. They usually can be kept in culture for about a week. Yeah, but and then there is another step when actually the gut organ is being modeled with a whole diversity of cells and epithelium. Yeah. So, but to answer your question, we do use all of the systems involved. Yeah.
Timur, another question. You mentioned that the whole revolution is like 15 years old. So, um, is it that microbiome studies did not exist or did not bloom 15, like 20 years ago? So what?
So the specific event that I'm referring to is application of what's called next generation sequencing or development of next generation sequencing and application of it to microbiome samples. So in terms of sequencing, the process of determining any gene sequence. So the first generation was kind of Sanger sequencing. So that's a very precise but first of all costly, second of all slow process where you identify each letter of the genetic code one by one, and that's by definition a slow process. So the next generation sequencing technology was a game changer for all biology. It's when we figured out how to massively parallelize this process. So basically take a long DNA molecule, split it into small parts, and then have them read or identified all at once. So this next step in technology, when it was applied to, well, it brought down the cost by several orders of magnitude, and this allowed application to microbiological samples. Have I answered your question?
Yeah. Thanks a lot.
Yeah. Okay. So just one, 2 to 3 terms I want to cover, and then we dive in. So one is a receptor. So it's going to be quite important. To function, an organism needs to exchange information between parts, and one of the primary ways it's done in biological systems is through the system of ligand and receptor. And what it is that it's essentially a molecular detector or machine that went through random movements. Another molecule that is called the ligand comes in contact with this molecule called the receptor. The receptor then changes its structure. It's called conformation, and this event can then lead to a cascade of biochemical reactions, and for example, to us sensing certain smell or certain taste, or feeling happy or angry or sad or whatever. And this is directly tied to the theme that intrigued perhaps some of you in the beginning, that microbes affect our mood. And this is directly tied to the fact that microbes can pretty much produce any molecule they want, and that our cognition and our senses and our behavior are entirely dependent on this concept of molecules um, uh, getting to receptors and then leading to things. But we'll go to this in a bit. Another thing is antibody term. You of course already, everyone on the planet knows what about antibodies, but it's interesting and important to know that antibodies are essentially secreted receptors for foreign molecules. So there's no real difference. And with regards to receptors and antibodies, there was an interesting paradox in immunology a while back. So in terms of genes, we know that the genome of humans contains about 46,000 of genes, and only about half of that is protein coding, or codes, or is responsible for the matter, or like the machinery that we consist of. And yet our immune system is able to effectively recognize and fight whatever molecules we encounter, or whatever organisms we encounter, or at least learn to do that. And the numbers don't end up, right? So we know to recognize something, we need a receptor. To have a receptor, you need a gene. There are only 46,000 genes in our whole genome. And certainly the amount of molecules around us in the world is many, 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 many orders of magnitude bigger than 46,000. So how the hell does our
Sorry,
immune
I
system?
thought I've heard wrong the first time you said 46.
Yes.
Why do I think it's about 20 or something thousand protein coding? What are the 46,000?
That's because I'm including all genes. I said about half of that is protein coding. So 46 in total, about half of that, which is about 20,000 protein coding. So you're right. And but I wanted to make the point even stronger, that even if we include every single gene we have, we still cannot account for many orders of magnitude more things that our immune system has to come in contact with. And in particular, just an average human has or healthy adult person has about several thousand species of bacteria, only just the bacteria, and they produce many different molecules. So that's already outstrips that. So the answer is that antibodies and immune receptors that are responsible for recognizing foreign molecules, they are combinatorically arranged. So they're just a handful of immune genes that are responsible for forming a receptor, and then they are, in the process of forming a receptor, they are diced up, and then these pieces are randomly rearranged, and they form, of course, many more combinations. And then there is another process where a random sequence is just attached to it, and then there is yet another process in which different combinatorial combinations that arise compete against each other for the ability to attach to the ligand that the immune system wants to recognize. And then new changes are induced, or it's called hypermutation, where it's very similar to the way we teach computers to recognize pictures, or it's essentially a biological classifier with kind of positive and negative reinforcement, like neural networks that recognize your pictures on Facebook and or recognize that there is a face in the picture and so on. So it's not very different from that. And the last term I want to cover, it's antimicrobial peptide. So peptide is, of course, just a sequence of several amino acids, and some of them have bactericidal properties and can act as essentially small, weak antibiotics. Okay, so any questions up to this point? No, we move on. So the first thing I wanted to mention is okay, we know that we discussed the methods, how we know, let's imagine now that we took a sample of a human microbiome and looked at everything there. There are several things that fundamentally determine the relationship between us and this collection of organisms. That this relationship is very asymmetrical. For one, as I mentioned, humans only have 46,000 genes in total, about. But if we did what I said and we sequenced the whole genome of microbiome, then we will discover that the genome space there is about 30 million genes. And then if we compare two random people on Earth, while their own genome or human genome will be 99.9% identical, their microbiome genome can be as different as 90%. So from this, we can know first, microbiome has many more genes, several orders of magnitude more. That's one. And secondly, it seems that it's more diverse than our genome. And then if we do this experiment or this observation for several years in a row, and look at the dynamic, obviously our genome will also a little bit change depending on what you sample, but we can, for the purposes of this talk, assume that human genome is stable throughout an individual's life. But if we look at microbiome, we will see that on the overall structure, it's quite stable if we're talking about adults, but on like individual microspecies scale, we will see evolution on the scale of 2 to 6 years. And several subtypes and variants will arise that everyone is talking about now with regards to COVID-19. But the same process of new variants arising, of course, happens also with other microbes. And so this is another thing that is different between us and microbiome, that on the scale of our life, our genome is stable, but microbes' genome evolves and is much more diverse. What are the consequences of just the size asymmetry? It means they have much more biochemical capabilities that we do. They can eat more things that we can do. They can synthesize more things that we can do. And they can evolve quite quickly depending on the circumstances. So what this creates is an evolutionary incentive for us to use this ability of the microbes that we are associated with to essentially outsource certain biochemical processes. For example, any plant food you eat, any salad you eat, any fruits you eat, anything that contains what's called fiber, we don't have the tools to metabolize them at all whatsoever. And microbes do that for us in our gut. We of course know that there are amino acids that we need, and they produce that too, and they produce several vitamins that we require, and so there's this capability of these neighbors that we have, so they can provide us with small molecules that we couldn't get ourselves. They can extract energy from sources that we cannot access, and yeah, so this, for now, this too is enough. So then if we then look at evolution, this provides an incentive to enter kind of a symbiosis relationship. So the microbes get the food that we eat in the gut, and the shelter, and the stable temperature, and so And we get all these benefits of additional nutrients and stuff. But as in any relationship, you need to draw boundaries, and microbes will take advantage of us if we don't, or they will take advantage of anyone if you let them. And here's the tricky part. Our immune system is typically designed, we classically think of the immune system as this sterilizing force, or our shield that repels any infections if it's successful, and kills off bacteria, viruses, and so on. And it is true. If you have a small cut and bacteria come in, then in your bloodstream, then the immune system will respond and completely destroy any invaders. But then the question becomes, how does the same system with regards to our gut stay tolerant to all of this millions and trillions of bacteria that are right there? And the fascinating thing is that the thickness of our gut lining is just one cell. So it's just the single layer of cells separates our bloodstream from all of the trillions of microbes. And so it's very easy for our immune system to get overly active. And when it does happen, this leads to many types of diseases, for example, inflammatory bowel disease. So we know that this is not happening normally. And also there is another asymmetry. If we're talking about the kind of a military confrontation between bacteria and us, the forces are not equal at all, because in terms of numbers, the immune system only constitutes only 1 in 100. So every 100 cell in our body is an immune system. But there are microbes, approximately the same amount of microbes in our gut only as the whole body. So microbes outnumber immune cells 1 to 100 at least, and actually more usually. And if you think maybe, but maybe you don't have to think about it, but essentially also generation time is not in our favor. Our cells divide with our immune cells divide with a frequency of about 24 hours. They can divide faster, but essentially not an order of magnitude faster. But microbes can divide as fast as 20 minutes. So if you think you are outnumbered 1 to 100 and your potential enemy can replenish their forces or expand them several orders of magnitude faster than you, and they have more plasticity because they evolve and you do not, and also they have more genes than you do, so they can have more capabilities than you do. This is a very uneven battle. And our immune system in the gut is a very interesting system, because it overcomes this and manages this uphill battle with several quite ingenious adaptations, such as our immune system actively the main thing is that it actively uses microbes against themselves. So it promotes, it rewards and supports some of the microbes that are either beneficial to us or are not harmful. And then they, through competition, take up the space or the niche from other bacteria that can potentially hurt us. This concept is called colonization resistance. Is that it is based on the idea that the amount of nutrients or the amount of space physically inside the gut is limited, and it can be either taken by good bacteria or neutral bacteria, or bad ones. And if it is taken up by good ones or neutral ones, then it's not taken up by the bad ones. So divide and conquer, essentially, by our immune system. And also,
how does immune system promote this colonization of good bacteria versus bad bacteria?
Yeah. So that's a very good question, and there are many ways. I will give two examples that are most fascinating to me. One is in adults, and the other in small children. In adults, what happens is so one other asymmetry between us and microbes is that we have the ability to store energy and they don't. And also that in terms of life, for them 20 minutes is a new generation, but for us 20 minutes is not. What we know is that when we starve or when we are ill and we don't eat, the epithelial cells and gut lining start actively secreting proteins that are covered in sugars, special sugars, into the gut lumen, and these sugars are such that they can only be eaten by good bacteria that are supportive to us, that they provide us with small molecule nutrients that we need, and they stimulate our immune system. So you see, as I said, the immune system divides and conquer microbiome because otherwise it would just lose. And there is a feedback loop. So the immune system, for example, in this way, nutritionally supports good bacteria. And then this bacteria in turn secrete molecules that stimulate the immune system. So this is like a cycle of two partners supporting each other against the common enemy, which is all the other bacteria. And another example is, of course, the human milk. So when a mother nurses a child, we naively think, we may think that this milk is for the baby itself, but in fact, more than 90% of it, the milk is not for the baby. The human genome doesn't even contain the ability to metabolize most of the sugars in the human milk. In fact, human milk, most of human milk sugars are acting as prebiotics for the good bacteria to colonize the human child. So the mother has the instinct to kiss a baby and to be intimate with the baby, and this is all designed. Of course, it has many other biological and psychological, and also it's who we are. But another reason for this instinct is to transfer microbes, the beneficial microbes, to the infant, and the infant's gut will already contain the nutrients or the milk of the mother that is essentially a selective media. It is essentially the media that can only support this good bacteria and give them an edge over competitors that do not have this selective media. They cannot use this. So that's another example.
Can I jump in with not one
Sure.
but two questions here? So one is how well does my body regulate which bacteria proliferate? In particular, when I buy probiotic, capsules, some of them have whatever 5,000 strains, the other like 50,000 strains. Assuming that I just eat whatever there is, and then my body will choose the ones that actually match. So how well does the body do that? Do I need to care?
Yeah. So one thing, there are many compound questions, and it's this very important and interesting question. One thing about probiotics you need to know as consumers is that while it is true undeniably true that our health rests upon the health of the ecosystem inside us, just in a similar manner and to the same extent that we depend on bees and polar bears. But you also need to know that probiotics are not regulated like drugs in North America or anywhere else in the world. FDA does not approve them. FDA does not test them. In fact, my laboratory, previous laboratory in Israel, tested the top probiotics on the market in the US, and there is no guarantee. Essentially, there's no guarantee that what is inside can even survive or is alive to begin with.
Which laboratory at the Weizmann Institute was that?
It was the Elinav laboratory. Yeah, so you can look it up. It was a very high-profile publication because probiotics is right now a big industry, multiple $1 billion a year only in the US, and it's only growing, and it's going to grow because it is true that microbes determine our health to the great extent. But the regulation is lagging behind, and probiotics that you can buy over counter or in the grocery store know that there is no guarantee of whatsoever of anything. Yeah. Now, how our body selects bacteria. So there are many, many theories towards how the composition settles down. So one theory is, of course, as you mentioned, provider selectors. So selection of providers, so that we like eat whatever we want, and then there's like a stream of bacteria in us, and then our body selects what it wants. Another theory is that the host is kind of, or we are kind of passive, and there is just competition, kind of like a marketplace competition between bacteria inside us, and the most prominent win. And in reality, it's kind of in between, because there are evidences for and against both theories. So as I said, if we pick two people at random on Earth, the similarity can be as low as 10% between them, and both people can be completely healthy. Therefore, it's difficult to say that on a genetic level, we are programmed to select some bacteria over others, even though there are examples of that. For example, like the milk, like human milk, as I mentioned before, or the support during starvation, as I mentioned before. There are specific examples of that. But we also see that two healthy people can have different, 90% different microbiome. So the extent of how many bacteria are actually kind of coded into us, or we are programmed to support them, is not definitely not all of them. But also, in terms of the competition theory, is also not cannot describe cannot describe or explain the succession of compositions that we go through. So in life, first we start life sterile, and then through skin contact with the mother, and through the delivery, and through other means, there is colonization, directed colonization of the newborn. And interestingly, there are milestones in terms of microbiome composition, in terms of, that is very similar across all people. So if there is just random competition, this cannot explain that. So I will repeat again, because maybe it wasn't very clear. Babies go through a succession, a quite conserved succession of bacterial communities that colonize their gut with every year, up to year 3. And between babies, there isn't that much difference. If you if you compare their composition. So at the moment, we don't have a unified governing theory of how the diversity of microbiome arises. We are working on that. But with regards to mechanisms of how our body selects, there are several other mechanisms. As I mentioned before, there are these nutritional benefits that microbiome can provide us, and also, as we mentioned, through colonization resistance, through occupying the space that a bad bacteria can occupy, they also protect us indirectly. So all of these things create an incentive for our immune system in evolution to develop means to not only support the good bacteria, but also actively deselect the bad ones. And this is why in the beginning I wanted to cover the term antibody. It's because we produce on average, adult people produce about a milligram of antibodies in our gut, which is a huge amount of antibodies every day. And these antibodies are targeting specific bacteria. So I will repeat again. So our immune system competes with itself and works hard to be able to recognize certain bacteria in our gut and tag them with antibodies to eliminate or to promote. Actually, it's not yet clear. For some reason, we don't know yet why. Tagging bacteria with antibodies for some bacteria is beneficial, for the others it's detrimental. The mechanisms, as I mentioned, is unknown at this point, but you need to know that production of antibodies, especially at this scale, is very costly, metabolically costly, and it occupies the capacity of the immune system to the great extent. But the net effect of this curation with antibodies, very targeted species-specific curation, is supporting the diversity of microbiome. Because if you think about ecology and competition, the most efficient stability, diversity, and efficiency of a system are not they don't work in unison, and we know that, and we feel the consequences in the pandemic. We as a society, as a race, were striving for efficiency and developed this very complex and efficient chains of supply chains, and they were very efficient, and they are very efficient. But what they are not, they're not stable, because efficiency and stability are two sides of the same coin, and they trade off. So stability, if you, in any system of any kind, doesn't matter whether it is firms, individuals, or bacteria, if you have cooperational interactions, it increases efficiency, because you cooperate, so you don't both have to spend resources to achieve the same task, and you achieve it more efficiently. But what it also creates, it's dependency, and of course, when the time comes when whatever pandemic comes in for us as a race, or you decide to go vegan, or the other way around, you stop being vegan, for the microbiome is an apocalypse. It's a switch of all the nutrients that you take in, and this is a massive stress. And so bacteria that are codependent on each other, they will all fail and go extinct potentially. So that's one thing. Another property of systems is diversity. And if there is no restriction on competition, the number of species in our gut will be roughly equal to the number of nutrients we put in, because for each nutrient, if there is unrestricted competition, just one will win through evolution or some whatever, and the others will perish. And so roughly the number of nutrients will be equal to the number of species. But that's not what we observe. We observe the number of nutrients we take in is on the order of 100, several hundred if you eat very healthy. But the number of species of bacteria in a healthy person is like 3,000. So there's a mismatch. And this is where this antibody secretion in the gut comes in. Our immune system actively dampens the competition. So our immune system not only divides and conquers, but it also acts as so it doesn't let the strongest bacteria win in the competition, because then the diversity of microbiome will be severely reduced by several orders of magnitude. And this is not what is beneficial to us as to humans, because we rely on this 30 million genes that microbiome has to provide us with all the goodies and metabolic goodies. If the microbiome will shrink in its diversity to just the most efficient, the strongest few hundred, next time you switch diets, everything will be lost, and there will be a catastrophic failure in ecosystem in our gut. And as a result, we will face depression, our immune system will fall, we might develop diabetes, we might develop coronary disease, we might, many, all systems, our sleep can be disrupted. So
can I ask a question?
Yeah.
Since Zhenya Shapiro stepped out, I will play physicist here. So you, oh God, he's back. You keep saying things like this production of antibodies to kind of modulate the balance of good bacteria and bad bacteria is very costly. I wonder if you could throw some numbers for illustrating sort of the life cycle of this, how costly, what do we compare this cost to? If we produce an antibody, what's the life cycle, how long this particular molecule hangs around, is it going to stick to some bacteria and get out of the system? And sorry, there's a second part of this question. What I always found fascinating is how rapidly a dead organism is run over by bacteria, right? It's just almost instantly in front of your eyes, a dead fly, a worm, if you're watching under a microscope, while it's alive, maybe the heart is beating, but almost as soon as the heart stops, it's overrun by dividing bacteria. I always puzzled why, which processes stop and what changes in this balance. Maybe you could comment on that.
Yeah, so it's very good you called me out on what that exactly does efficiency or cost mean. I cannot provide you with all the numbers that you're asking for. Probably they are out there, but what I referred to as cost was not directly like units of energy that or calories that are being spent. What I meant is every time your body decides to produce an antibody, it means there is a clone of cells called B cells that actively secrete this particular antibody, and they can only secrete this one. And they need a place in your bone marrow. And your bone marrow, it has physical limitations to how many clones and cell types it can support. Therefore, what it means is if you are, our immune system can recognize anything, essentially. Its plasticity is, it can just develop receptors and antibodies against anything, against Teflon or whatever, it doesn't matter. But at any given time, the capacity of what it can recognize at this moment is limited to the supporting capacity of bone marrow, essentially. So if you're recognizing hundreds of species of bacteria in your gut, it means you're not recognizing something else. So that was the cost that I was that I meant. Of course, there's also, yeah, there's also
I see, not the protein synthesis cost.
Of course, there is that too, and 1 milligram is a lot. It's comparable to all other antibodies that are in your body everywhere else combined. That's what I mean. If you think about ourselves, like the way we interact with the environment, we essentially interact with the environment mostly through our gut, because our skin is essentially a barrier. It's not a kind of it's not an interface. It's the job of the skin is to keep things out, not to interact or exchange stuff. And our lungs are don't have many microbes in them. They're very few. So in the main way we interact with our environment, as I mentioned, is through the gastrointestinal tract. And when you look at this way, then it's not surprising that our immune system devotes half of all of the antibodies it has, half of all of its defenses to this frontier gate that we have. And this is mirrored not only in antibodies or B cells, but also in all other immune cells. So our gut is our biggest immune organ. It has as many lymphocytes as the rest of the body, but it's also our biggest sensory organ because it has the same amount of neurons as our spinal cord. In fact, among the peripheral nervous system, so we have the central nervous system as our brain and spinal cord, and among the peripheral system, the gut is the biggest part of the nervous system. It's called the enteric nervous system, and it's almost autonomous. Like our body can survive if our brain is dead, but if you kill the enteric nervous system, our body cannot survive. And the enteric nervous system, so there is an operation called vagotomy, which severs most of the connections between the central nervous system and the enteric nervous system. And enteric nervous system does fine. It can operate essentially by itself. And one of the big revelations of the last few years, not 15 years, few years, is that a gut-brain axis exists. What I mean by that is that bacteria directly produce neurotransmitters. Neurotransmitters are molecules that are used by our brain cells to communicate to each other. So they form patterns of thought, memory, and there are several kinds of them. And for example, one of them is called serotonin. Perhaps many of you heard of it. And maybe you'll be surprised to learn that 90% of the serotonin in our body is produced by gut bacteria. And there are many other neurotransmitters that are produced by the gut. They are then uptaken by the enteric nervous system, which, as I mentioned, is the size of our spinal cord. And then all of this goes to our brain. For example, so what do we know? We know that there are many. We cannot yet directly state that this or that neurological disease directly is caused by microbiome, but we can already now say that we certainly see many diseases differently now. For example, autism. The scientific community now recognizes autism, that autism has a gastrointestinal microbiome component. The reason for this is that two-pronged, animal studies and human studies. So with regards to human studies, because they're of course more more relevant, but less in number. So there are just a few cases where autistic or not, like children that later became diagnosed with autism were not diagnosed before. They underwent several courses of antibiotic treatment and antibiotic for unrelated reasons to autism. And of course, this audience doesn't need a reminder that we as a society, totally overuse antibiotics. And for the whole paradigm that I'm now building, where we recognize that there are good and bad bacteria in our gut and that we evolved specific mechanisms to protect and cooperate with part of our microbiome, and that we rely on this microbiome for our health, then the concept of antibiotics as an indiscriminate weapon of mass destruction, when we're talking microbes, becomes less appealing. And as I said, for example, many neurotransmitters that we need produced in our gut, and when you wipe out repeatedly all of the bacteria, regardless good or bad, there were several cases in which this severe use of antibiotics was followed. I cannot say it was caused, but was followed by diagnosis of autism. Now with causation experiments in mice. So how can we say that the bacteria plays a certain role with causality? The way we do this is we have animal models that don't have any bacteria whatsoever ever. They're called germ-free mice. They live in like space kind of isolators, so they air gapped everything. You have to wear the whole suit and so on when you work with them. So this bacteria never in their lives seen a single microbe, not a virus, not a bacteria, nothing. Their parents haven't for many generations. And then we can compare them to their siblings that are genetically identical, 100%, that are not germ-free, that do have an association with the microbiome like we do. And then we can perform different types of tests, for example, on memory and on social behavior, because mice and rodents like rats especially, are very social and very they have high cognition. And of course, here we're talking about, in the beginning of this whole lecture, we mentioned that when we're talking about animal models, they are models. And so when we ask a question, we need to phrase the question in a way so that the answer would be applicable, would be so fundamental that the answer would be applicable to both us and the rats or mice. And
Can
when
I ask about this germ-free mice?
Yeah.
I'm having a hard time imagining how you could possibly have germ-free mouse. I can think of maybe other organisms, but a mouse is born and it gets a microbiome from the mother, and so it's a chicken and egg problem. If you need to have a germ-free mother to have a germ-free, yes, you can purell the hell out of environment, you can microwave food, you can filter the air, but how does it not get bacteria from the? How does the first ever germ-free mouse not get bacteria?
So there are two ways. One kind of an old way, now a new way, which is better. So the better way is essentially IVF. So
I didn't catch that. I'm sorry. What is it?
So you one way is you do a C-section essentially for the first animal, and this procedure is not has a very low efficacy rate because of all the so the contamination rate is quite high, but you can succeed rarely you can succeed. And then after that, what you do is you work with you do an IVF essentially. You have a fertilized egg, and then this egg you transplant into a germ-free surrogate mother, and this is how you then propagate. So the answer to your question is yes. The first step is very inefficient. You do need to succeed a few times out of many, but then this can be propagated with the use of fertilized eggs and implantation into surrogate.
You can do a C-section, but then you need to wash the gut with ethanol or something. Anyways, that's not too important. I was just curious. Do these mice live, the germ-free mice? What is their life expectancy compared to normal mice?
They live pretty much the same. They don't. We don't see them dying off young or whatever. But they have many biological differences. So one of them, as we started talking about gut-brain axis, is in their behavior. And I've been saying that when it comes to neurological diseases and disorders, it's very difficult to justify, comparing disorders in rodents and in humans. But there are certain things we can do. For example, we can test their memory, and we can test how social they are, and we can test anxiety. Because, for example, anxiety, we can just check there are several hormones that are responsible for the feeling of anxiety. We can just directly check their levels. And also in terms of behavior, there are certain behaviors in mice that are indicative of anxiety, and this is well proven because we can measure anxiety directly with the hormones, and so what we've seen is that germ-free mice display kind of an autistic behavior. So they are much less sociable. They have problems in memory as well. On top of that, how can you test the mouse memory? For example, you can either use a maze and measure the average time that it takes a mouse to pass the maze. You can also introduce objects and measure the time that mouse is sniffing and exploring the object, and then introduce. Let's say you put in a cage two objects, two different objects. For example, a sphere and a cube, and mice. And measure how much time mice spend exploring each of the objects, and then you switch one of the objects to some other shape or whatever, smell or whatever, and measure again. And a mouse that has microbiome will spend much less time exploring one of the two objects that they already met before or explored before, and focus its attention on the new one. But the germ-free mice, and this has been replicated around the world many times, so it seems to be true, the germ-free mice explore equally, spend the same amount of time exploring the object that they before saw already, and the new one that they have never seen before. And in these types of tests, and in tests in Labyrinth, we see that there is a strong impairment in memory in germ-free mice. They also, we see microbes affecting their behavior and their risk-taking behavior. So mice that are normal mice with the microbes, they usually avoid open spaces because they in nature are associated with predators like owls and whatever. That mice do not like open spaces, being in the middle of the road. They like being close to the corner and defended at least from one side. And germ-free mice have a problem with sticking to this pattern of behavior. They go into open field. This is called an open field test. And if you measure the time that the mouse, spends in the middle, you like put a camera and you record many hours, and then you analyze the footage and quantify it and so on. And you can see that germ-free mice take much more risks than SPF mice. And
can I jump in with a way lower level? I probably have been watching too much science fiction lately, but basically what you're describing. So if I can say that you will figure that one type of bacteria produces one type of neuromediator or something like this, the reality that I'm hearing is that by mid-century, like 30 years from now, I will be able to buy a food additive that will make me a better mathematician and uh, make my memories better, or make me a better lover maybe, or uh, turn me from a mathematical type to a painter type, right? So
to the extent, I'm not sure this is the extent of what might be possible. Certainly, what I can see happening is it is probably true that depression has a component in the gut component, and that could perhaps be solved with as you said, in several years, with either probiotics, or live cultures that you take in as a pill. We already have several therapies that are working like this. For example, the very famous example that everyone always brings up, there's this infection called C. difficile infection. It's a bacterial infection that is lethal to immunocompromised patients. And this C. difficile bacteria is resistant to many antibiotics that we have. And anyway, we're running out of antibiotics, so we are screwed on that front. Pretty much nothing we can do about it at this point at the current state. But C. difficile is not being a problem because we figured out that the bacteria that are the bacteria that effectively compete with the difficile and decolonize it. So if you have an infection with the C. difficile, there are very strong. Yeah, it's a solved problem essentially. You take a consortium of several bacteria that are designed in a way to outcompete C. difficile, and the C. difficile goes extinct because together those bacteria outcompeted, competing for nutrient sources. Sorry, you're on mute, I think.
It's good. Yeah, thanks.
Yeah. Another other therapies that we have at the moment is they are immunomodulatory. So as I mentioned before, there is this positive feedback loop with good bacteria in our immune system, that immune system supports good bacteria and good bacteria support the immune system. So there are now this, and the laboratory I'm working at now is actually responsible for developing several of them. So there are defined mixes of several bacteria on the order of 13, 20, something mix that you take, and it promotes the development and the division of your immune system in the gut, and basically strengthens your immune system essentially. So that's already available now. Yeah, but with regards to neurological diseases, it's more difficult to quantify. Yeah, so it's a more it's more difficult problem, of course, than the immune system. Sorry. Any questions?
I'm not sure because we don't have slides, so I'm not sure how far we are from the end of this session. But I have some practical questions, and I'm kind of wonder if you continuing with the theory, or should we ask questions like?
Absolutely, please ask away. That's the reason why we don't have slides, because I wanted to follow any interest and questions you have.
Okay, so from practical standpoint, from what I heard, there are several things that are very important in the initial phase of the beginning of the life. It seems like being born in a natural way, at least based on what you said about mice, beneficial for us, and having the mother's milk in the first phase of your life. It's also beneficial for microbiome and for our ability to produce all of these interesting immune properties. Like, let's say later in life, what can we do? One thing, what I heard is the diversity. It's obvious. I think that having a diverse kind of diet and all of these things are kind of beneficial for us. Anything else? Anything else we are missing, like from adult standpoint? What can be done to help our microbiome?
Yeah. So I'm not gonna. There are no big revelations you don't know. What our parents have been telling us, that eat your veggies and eat as many as possible, is true. It's just that now we are learning why, and now we're learning how to fix that if we didn't do that. So the thing is that we now do not have a precise definition of what constitutes a healthy microbiome. We know of several properties that it must have, but we don't have an all-encompassing theory of that, because there are many. The inter-individual differences are very big between many healthy people. So, but things that we do know, one, as you mentioned, is diversity, and one way to promote it is to eat as many different types of fiber as possible. So different vegetables, not the amount. So not like a lot of the same salad, but as many different plants as possible is much better than just, yeah. So that's one thing. And another thing is, not surprisingly, exercise. So microbiome is very sensitive to exercise, and there have been studies where you take microbiome from an exercising person, transplant it into germ-free mice, and they have benefits to their health, to their metabolic health, immune system, and so on. You transfer microbiome from a sedentary person, a potato couch, into a germ-free mouse, and it becomes it displays problems with metabolism, especially metabolic syndrome, and their immune system is reduced, and their sleeping pattern is not as sharp.
Timur, I'm sorry, but you just told me that completely germ-free mouse lives as long as the ordinary mouse. So why do I care to help my microbiome? Seems that it's useless, other than maybe good mood and serotonin.
No. So you need to understand that there is a limitation to you need to. So one thing is when I say I was inaccurate before. When I said there is no difference between the lifespan of germ-free mouse and SPF mouse or regular mice, it means within the time tested and within the grants that were granted to study this problem, which is not, so lifespan of mice is several years, and germ-free mice can, so the lifespan is like about 2 years, and the longest germ-free mouse experiments I've seen are like 1 year. So no one has ever, to be honest, so no one has ever maxed out the lifespan of a germ-free mouse, but within the 1 year, we didn't see like a huge drop of, in mortality.
Okay, before the question police shuts me up, I also want to ask if you know what happens to microbiome on rapamycin.
Well, rapamycin is one of the antibiotics, so it will target. What do you mean? They die?
So rapamycin is the only drug that is known to prolong sort of natural lifespan and health span in mice and most other species. It is also a massive immune suppressor, and so that's why I'm curious if you knew, as rapamycin
So
suppresses immune system and prolongs life, what happens in the meantime to the microbiome? But I guess maybe the study hasn't been done yet.
So actually, right now, like a laboratory that I'm at, we had a Nature paper accepted. That's going to come out on the. I'm in Japan, so what the benefit of that is that there are many centenarians here, people who live to 100, and the forms that you fill in to whatever to get your mobile phone, whatever, they have 3 digits in the age part. And we studied a microbiome of centenarians, and it's different from people who don't live to that age, and there is a component to in microbiome and longevity. However, I cannot speak to the specific effect of this drug that you mentioned. But all of us have taken antibiotics, antibiotic courses, and if you want to know how resilient microbiome is, it is quite resilient. So you take an antibiotic, even if you take a combination of antibiotics that target pretty much all, because antibiotics are not equal, they're antibiotics that target only some bacteria and not others. But even if you take them in combination to kind of cover everything, the best you can do is about 90%. Even then, in a space of a week, your microbiome will rebound. So you take a course of antibiotics, even combined, then you stop. You give yourself a week. If you then continue the same diet you had before, within a week, your composition will rebound to what it was before your antibiotic course. The problem becomes if you do multiple courses in a row, then we see that then the microbiome does not recover, and then many beneficial species go extinct, and in some cases it leads, to certain diseases.
May I ask a question?
Sure.
You said that there is no surprise that exercise helps my microbiome. For me, it's like a huge surprise. Like I have no idea how would it actually benefit the microbiome. Can you explain?
Yes. So microbes.
Can you repeat the question? Sorry,
Yeah. So there was a statement that the exercise helps the microbiome in general, and it's like it's not surprised. I'd like to know why it's not surprised and how it does benefit the microbiome in general. Just exercise.
Yes. So so the reason why it's not surprising is because I was previously saying that eat your veggies. So the next cliche would be to exercise to have a better life, and microbiome is a mediator of that, partly a mediator of that benefit of exercise. So that's why it was like I was saying it's not surprising, eat your veggies to exercise. Anyway, the reason why there are many mechanisms. One of how exactly microbiome exercise helps the beneficial bugs in our gut. One of them is through regulation of oxygen metabolism in our gut. Essentially, normally our gut doesn't have any oxygen. It's anaerobic. And but what separates good bacteria from bad bacteria is that among bad bacteria, there is a higher percentage of what's called unobligatory anaerobic bacteria. So they can live with oxygen or without it, as opposed to good bacteria that are exclusively anaerobic. They get exposed to a bit of oxygen, they all die. So as I mentioned before, what separates us or our bloodstream that is very rich in oxygen from the gut, from the bacteria in the gut that have, most of them are very beneficial to us, is just one cell lining. And molecular oxygen, it would, with no adaptations, it would just filter into the lumen and provide the environment for pathogenic bacteria to live there and ruin your health. What happens in people who exercise is that their gut, the epithelial cells in your gut, they start consuming a lot of oxygen, and it never reaches the gut. Essentially, it creates an anaerobic environment supporting the good bacteria and also at the same time preventing the bad ones from proliferating. And an interesting thing is that the good bacteria also, so the good bacteria we're talking about, eat your veggies. So they metabolize fiber, and the byproduct of this metabolism, one of the byproducts is called short chain fatty acids. And this is like the big buzzword in the medical world right now, because they do a lot of good things for us. One of them is this short chain fatty acids. They are then uptaken by our epithelial cells of the gut and can be used as fuel for burning oxygen, thereby even further reducing the leakage of oxygen from our bloodstream into the gut. So not only our exercise helps this process, but also this bacteria, that beneficial bacteria that need it to not have oxygen, they actively try to help themselves to, to stop this leakage.
I think that's amazing and surprising. Probably I have a couple more questions. I'm sorry. I hope we have time to address them. So what do we know about the effect of the diet, not the diet, but the fasting on the microbiome? Like, how far we can take it? Like, if we have any studies on like the longitude of the how long we can do that, and it's still kind of not harmful for our microbiome.
So if you're talking about prolonged fasting, as we mentioned earlier briefly, like for several days, then what happens is that our body recognizes that while we have some fat reserves, the microbes do not, and we do not want a catastrophic failure of the ecosystem within us because we depend on it. So the epithelial cells in our gut start secreting food for the good bacteria, that only good selective food that only good bacteria can eat. That's one thing. If you're talking about intermittent fasting, like on the order of 12 hours or something, then it's very, very beneficial to both your microbiome and you metabolically. And studies have been done. I myself have done studies on this. It's because our microbiome, as we do, has circadian rhythm. So if you sample our microbiome at equal, every 4 hours or equal intervals of time, the composition changes, and actually you can backtrace, you can tell time based on the composition of microbes. And so they have that's of course because as we do, as all organisms, need to resolve different biological processes that are not compatible. So for us, the problem is easier because we are multicellular organisms, so we have different organs, so we can separate different processes better. But still, we do still need circadian rhythm to have opposing biological processes not crash each other and to increase efficiency. But for single cellular organisms like microbes, it's a crucial thing to divide at one time of the day, produce toxins to kill your competitors in the other time of the day, and the other time of the day metabolize some nutrients and so on. Therefore, when you constantly eat and nibble, you disrupt that, and so it disrupts that, and it leads to not healthy microbiome, and therefore everything else is all the benefits are reduced. So 12 to 14 hours, great for microbiome.
Thank you. That's very interesting. And I think the last question that I have is, what excites you the most in the current development of this field?
So the exciting thing is that as I mentioned, microbiome has several orders of magnitude more genes that we do, and microbes can evolve to do pretty much anything. And we co-evolved with them for so long. We only now recognize microbiome as the forgotten organ that is connected to any and all of our systems. That the potential to promote and improve our health, learning about microbiome and using microbiome to this goal, I think I don't see the limit to this potential. So I'm excited about utilizing metabolic capabilities of specific microbes to solve some health problems that we cannot solve at the moment. For example, we close to 80% of all drugs we as humans have, we take orally, and as you and it's very easy to put two and two together. That on one hand we have microbiome that has this almost infinite metabolic potential to metabolize anything, to synthesize anything, and on the other we have all the drugs that we take in, and they all converge in the gut, and it is known that microbiome can and does metabolize many of our drugs, and so there are certain strategies that we can employ to decrease loss of efficiency, decrease waste. There is a point to be made that we are currently wasting a lot of our drugs and deploying them inefficiently, and there are many there are many strategies where we can, for one thing, for example, we can reduce waste. We can, by eliminating or editing the composition to exclude one way or the other bacteria that deactivate the drugs we take. Or alternatively, we can now do the other thing there. We can have bacteria activate an active compound of the drug and boost efficiency. Or another strategy would be to have the bacteria to, producing what we need at a steady level, rather than having enormous peaks in concentration and then withdrawal, and then, not withdrawal. It's not all drugs are addictive, of course, but biologically having a steady concentration would be much more preferable to huge fluctuations.
So not a question, but an idea that I'd like to share, or maybe the question: did anybody try to face this problem of antibiotics being almost ineffective, or they will be ineffective in the near future? Did anybody try to use the those bacteria against bacteria, basically trying to understand what kind of bacteria causing the problem, and use other bacteria to attack them instead of killing everybody?
Absolutely, and that's not available right now, but it's going to be in a few years for sure. How I know, because we're working on it, and it's much more effective. I can tell you that it's much more effective than antibiotics. Not only because antibiotics do not achieve full decolonization, they're typically at max 90% efficient, and they are not targeting. It's a weapon of mass destruction that is not very efficient at that. But using microbes against microbes, as you said, in a competition is a natural way, we are working at this right now, and they're promising results. So we can essentially, it's a more efficient and more targeted. It's just better on all fronts, and you can the basic theory behind it's a bit difficult, but what you do is each bacteria has a set of nutrients it can metabolize and a certain competitiveness for each nutrient. And so what you want to do is to assemble a dream team of other bacteria that would all efficiently compete with this bacteria you want to decolonize at all of these parameters, at all of these nutrients, and would together overcome the competitiveness of this bacteria on all parameters. And this leads to complete extinction of this bacteria. And that's the future, essentially. That's what we'll do. And we'll not use antibiotics, of course, we'll use in operation rooms and in other places, but taking them for some gut flu is insane.
So in a sense, that's a good thing that we will not be using antibiotics soon, right? So we'll have to come up with new ideas, and they will be better than what we are using right now.
Definitely, yeah.
Timur, did people figure out about, let's say, which bacteria produce serotonin, and are there any research to grow this particular bacteria, let's say, to multiply it and put it back in the patient that has, for example, depression or some other mental problem, and stuff like that, or for some other as well indications?
Yes, so very good question. And yes, we do know several examples of bacteria that are members of normal healthy microbiome community that are responsible for production of serotonin. We cannot say they are the only ones that do that. We also know of other examples, not only serotonin, but also dopamine, for example. Of course, dopamine is, of course, the hormone responsible for motivation and the sense of pleasure. You get it after exercise and other activities. And boosting dopamine would a lack of dopamine is of course the reason behind Alzheimer's disease, and 70% of Alzheimer's patients also this early onset, many years before 70% of patients get eventually diagnosed with Alzheimer's. Many years before they start experiencing gut, intestinal problems, and so coupled together with the ability of bacteria to produce, but also lower the bioavailability of dopamine, led to the theory that Alzheimer's disease can also have a gastrointestinal component. So at least we can use it as a diagnostic tool meant to flag people at risk many years before they can potentially be diagnosed. And we do know of examples of bacteria that produce dopamine, or the other way around, reduce efficacy of the Alzheimer's treatment. And right now, the drugs are not the drugs. The formulations of bacteria that do this are not available, but it's in the works. We're working on it. Many groups are working on it, and I think there is potential there. We might see exactly this type of treatment, the bacteria that would. So we now know that bacteria heavily contribute to the brain chemistry that we have. Therefore, it's conceivable that we might figure out a way to change brain chemistry in people with disorders, not through chemicals, but through colonization of their gastrointestinal tract with bacteria that would constantly produce it at the normal level. It's conceivable. I hope so.
I should tell you that I know a guy who runs a hedge fund and claims that somehow his performance is so much better when he takes augmentin that he just periodically gets a week long course of augmentin to stay sharp with all with all the modulators, all the amphetamines, and God knows what available. And of course, this is just one crazy guy, but it's kind of a curious anecdote.
Definitely, yeah.
You keep referring to good bacteria and bad bacteria. Everybody understands it's just a metaphor, but I also noticed that when you say circadian clock, you surprised me because I expected to hear about some clock within bacteria in terms of gene expression, protein modifications, but you actually meant periodic changes in proportions in bacterial populations. So I'm very curious about changes within bacteria. Do you know cases of flip-flopping bacteria, which are good or bad depending on some kind of a context?
Absolutely, of course. Yes, it's an oversimplification. So regarding the circadian rhythm first, and then to the flip-flopping of bacterial roles. First, we are, so in nature, there are examples of bacteria that do have internal circadian clocks. And the adaptation of separating your space or your internal space in time as well as in space in different organs or cells is very ancient. So it's crucial to life. It's such a boost in efficiency that pretty much all have it. In the gut, we haven't. There are no reports yet that the clocks in the gut bacteria have been described. What we do know is that both on the level of community structure, they fluctuate, as you said, and also on the level of transcriptome, or on the level of what they do, they also separate their tasks. It is driven, the causation experiments show that it is mostly driven by our circadian clocks. So mostly by our feeding behavior, and that's why nibbling is very bad, because it ruins their cycle. If they had their own clocks, they probably could have perhaps overcome it, or be better protected against this. But it seems like the main driver of this fluctuations, both on the compositional level and on functional level, is our clocks. Now, with regards, yeah.
Sorry, I just want to say that there are not too many kind of very striking results when it comes to health and biology. And one from my personal experience, you could say, is I knew several people who were, let's just say, morbidly obese. And I had this experience when, in the course of maybe a couple of years, several episodes when I met the person and I couldn't recognize them because, they became just drastically, skinny, and
But then they rebounded, right?
no, that was all the same answer. Like, whoa, what happened to you? How come you lost so much weight? So a diet became popular when you eat anything you want, but only before the sunset. As soon as the sun is down, you stop eating. And of course, it just has an effect of limiting the calories, kind of implicitly. But I'm very curious.
Actually,
Yeah.
yeah. Actually, that's not the. It might have led to reduced calories, but I've done this experiments, not myself in mice, but also many other people have done it. Is that if you are to keep the same calorie input, if you just change the distribution of calories during the day, while keeping the same amount, the total amount the exact same, it has an effect on metabolic health and on weight. So if when you limit, there's this model in mice called Western diet, basically very bad. 60% of calories derived from fat, and this way we model McDonald's and fast food.
60% derived from fat.
Yeah. So this is of course a severe model, but obviously when you give it to mice, they rapidly gain weight. But the crazy thing is that if you limit their access to this very bad food to just 12 hours a day, they lose weight comparing to mice that have access to this food all the time and can eat whenever they want.
That's not so simple. I must tell you right now, I'm on a diet where 60% of my calories come from fat, and I'm losing weight because, again, nobody really knows how this works, but it's a diet where it's a low carb diet. So yes, I eat tons of fat and
ketonic diet.
and no carbs.
Ketonic diet.
Yeah, and I'm losing weight.
Yes.
But this is going too far into diets and stuff.
Yes.
Right. So this effect, going back to the effect of circadian clock and metabolism, I wonder if you really think that within the microbiome, it is the change in the population of bacteria from day to night, or is the change in some sort of enzymatic activity of the same bacteria from day to night that would make a difference?
Well, both happen. We can observe both, but coming back to metabolism, there's this work that's been done in my previous laboratory in Israel, and as a company was born out of this. The company is called Day2. Is what they found is, or what we found is that based on microbiome composition, two people, you and I, eat the same food, for example, rice. 100 grams of rice, the exact same, prepared the same, just then divided into two. And then we measure blood glucose, yours and mine, and we can expect very, very different results. So we have actually done studies on several thousands of people in Israel. And there are examples where one person eats a chocolate chip cookie and the other person eats rice yes, I think it was rice. And they have, you couldn't tell. You think you can tell by the glycemic, by the spike in the blood glucose, but you can't. So basically what I'm saying is that based on microbiome composition, how the glucose peaks in blood between different people can be very different. You and I take the same food, but for example, for me, there will be a high peak in glucose concentration in blood, which is a sign of metabolic syndrome and needs to be managed. But you having ate that food can have like flat line and no or a very small peak, and this is driven by microbiome differences, because you can transplant your and my microbiome into germ-free mice, and we have actually done these studies, and you can rescue it or reverse it, and yeah. So if you're interested, you can look it up. Day 2 glycemic response. Yeah. So yeah, that company essentially what it does is it profiles your microbiome and then makes suggestions of what changes you need to do to your diet to avoid these peaks or spiking of glucose in your gut, essentially. With regards to flipping to your first question, you asked whether some bacteria perform different roles in different contexts. Absolutely. So, for example, one of the good bacteria in most cases called Ruminococcus, what it does, so there is one of the strategies our immune system has to manage the overwhelming force of bacteria in our gut, being outnumbered, the generation time is much smaller for bacteria. They can replenish their forces faster, evolve faster, and whatever. So one strategy is to have a moat. So our gut doesn't have a direct contact with bacteria. Some epithelial cells secrete what's called mucus and form a mucus barrier. Mucus, it's like slime, but it's actually a very clever slime. It's like a net of, it's a molecular net that keeps the bacteria out and filters them out basically by size and excludes them. So there's this buffer exclusion zone between us and bacteria. But this net is made out of proteins and sugars of a certain kind that most bacteria cannot metabolize, but this Ruminococcus can. And essentially what this bacteria can do and does is metabolizes it, makes holes in our moat. But when it does that, it produces this short chain fatty acids that I mentioned that are beneficial to our immune system. So in normal situations, they are actually classified as good guys because they support our immune system. But when there is a catastrophic change in our bacterial composition, or for example, our gut is inflamed because we got poisoned or whatever, or something of this sort, then this foraging activity of this bacteria that it is eating our moat can be very dangerous, because it would just do its job that it's doing regularly, eat our moat, but then it can lead to other bacteria.
I don't understand this word you're saying. Eat our what?
Moat.
Mold?
Moat. It's so you have a castle, and around the castle there is water. So this is called the moat. or what Warren Buffett says, when you invest the company, it needs to have a moat, competitive advantage, and so on. So essentially, there's this barrier or exclusion zone between us and bacteria that is produced by our cells, and this barrier is being eaten by this particular bacteria. And normally the byproduct is good for us, but in situations when everything else is already bad, this bacteria can do it much worse, because then it can deprive us of this exclusion zone. And then what happens, all of the bacteria, good and bad, come in direct contact with our epithelial cells, and then our immune system freaks out because it is designed from scratch to exclude any and all bacteria from our body, from the bloodstream. And here it comes into contact with all of these bacteria all at once, and the immune system, the immune reaction starts, and it's more like an autoimmune system, because the bacteria that are being killed are also good. And this is part of what's called irritable bowel syndrome, and it leads to permeable gut, leaky gut, and leaky gut, it leads to systemic inflammation, promotes cancer, it contributes to depression, and it can lead also, we can see that in models of obesity as well. So in this situation, this good bacteria becomes a trigger or a tipping point between just a small, like a hiccup in the situation in the gut, and it transitions into an inflammatory bowel syndrome that is a self-sustaining inflammation that can drive, for example, cancer.
Thank you. Thank you very much. Very interesting.
I suggest as the questions, please. It's been 2 hours.
Yeah. So yes, let's wrap up.
Yeah, that we stop recording, and with that, Timur, it's super interesting. I honestly, on my side, I didn't expect that a lecture without slides can be so interesting. And anyone who wants to stay and ask more, if Timur is ready to stay, to wait for more questions. But otherwise, it's very good, and thanks a lot to you.
All right, it's my pleasure. Thank you for having me.
Thank you very much. It was interesting. Thank you.
That was amazing.
Yeah.
Thank you so much.
Yeah, so interesting.
Okay, the recording will be published in a few days. Many thanks, everyone, and thank you, Timur, once again. It was incredible.
Thank you very much.
Okay.